PTEN phosphatase and tensin homolog by Liliana Contreras.
Anissa Kanemoto.
And Amisha Singh. Okay, so first up we have the abstract regarding our enzyme, which is the PTEN enzyme, also known as phosphatase and tensin homolog. This enzyme is a critical tumor suppressor that plays a pivotal role in maintaining cellular homeostasis by regulating the phosphoinositide 3 kinase protein kinase B pathway, otherwise known as the PI3K ACT signaling pathway, which controls cell growth, survival, and proliferation. PTEN acts as a lipid and protein phosphatase, negatively regulating the ACT signaling through dephosphorylation of PIP3, a key secondary messenger. Dysregulation of PTEN activity is frequently implicated in cancer development due to its effects on uncontrolled cell division and survival. In this study, we primarily used Western blotting alongside the comparison of immunoprecipitation techniques to analyze PTEN isoforms and explore their functional roles in the PI3K ACT pathway. Our data demonstrates that both PTEN and its isoform were enriched by immunoprecipitation. However, neither was modified by common ubiquitin or ubiquitin-like post-translational modifications, suggesting that the regulation of PTEN may occur through alternative mechanisms. We also investigated the effects of PTEN loss on cell morphology and adhesion. Interestingly, while cells lacking PTEN did not exhibit phenotypic changes resembling those of trypsinized cells, a combination of PTEN loss and trypsin treatment resulted in significant alterations in cell adhesion and morphology, emphasizing PTEN's role in maintaining cell-to-cell interactions. Furthermore, we confirmed that PTEN suppresses angiogenesis, a crucial process in tumor progression, but inhibits metastasis, highlighting its dual role in tumor suppression. Our findings provide valuable insights into the complex regulatory mechanisms of PTEN and its isoforms, which contribute to cellular functions beyond simple cell cycle control, including metastasis inhibition and angiogenesis regulation. Moving on, we have the introduction of our PTEN enzyme. Like I previously stated, it stands for phosphatase and tensin homolog, which is a crucial tumor suppressor enzyme that regulates cellular homeostasis by dephosphorylating both lipids and proteins. Its primary function involves dephosphorylating PIP3, a lipid secondary messenger, thereby inhibiting the PI3K ACT signaling pathway. By suppressing this pathway, PTEN prevents uncontrolled cell division and can trigger apoptosis, helping to avoid tumor formation. In addition to regulating cell proliferation, PTEN controls cell migration, adhesion, and maintains genomic stability. It binds to the plasma membrane via homodimerization, which essentially means one PTEN enzyme binds to another and then binds to the cell membrane. PTEN's ability to maintain genetic stability further protects cells from the accumulation of mutations that could lead to cancer. Loss of PTEN function disrupts these critical processes, which can in turn promote metastasis. Thus, PTEN is vital in preventing tumor formation, and understanding its molecular mechanisms is key for developing strategies to restore its function in cancer cells.
So for the experiment, the researchers had a speculation that PTEN isoform is a type of post-translational modification, or PTM, of canonical PTEN. So for this, they initially used a immunoprecipitation test, but used a Western blot to analyze the results of the experiment. To test this, the Western was used to first isolate alpha PTEN, which is one of the two isoforms, beta PTEN being the other one, and to interact with different antibodies of different cell lines on the N and C epitopes of PTEN protein. The NIH3 T3 cells are commonly used as a model system to study the function of PTEN, and they allow researchers to observe how manipulating PTEN levels impacts cell proliferation, migration, and survival. From the blot, it is shown that alpha PTEN is recognized by this cell line. They also used canonical PTEN to interact with alpha PTEN, and it was recognized as well. And although HeLa cells are considered abnormal cancer cells, PTEN is still functional in these cells and therefore it was also acknowledged on the blot. However, alpha PTEN was not detected on the blot by the PTEN isoform because it is not found extracellularly, meaning that alpha PTEN is a vital part of the internal structure of the isoform and it is not a PTM. From this experiment, they concluded that the PTEN isoform is either a translational variant of PTEN or a product resulting from RNA alternative splicing. Therefore, PTEN isoform and canonical PTEN are derived from the same mRNA, and thus PTEN isoform is not a PTM. The discussion also describes how alpha PTEN isoform is localized to mitochondria, where it collaborates with PTEN to regulate mitochondrial energy metabolism. So PTEN isoform has PTEN-like activity to inhibit PI3K-AKT signaling pathway. This signaling pathway is responsible for cell growth, proliferation, and metabolism. And since we know that PTEN is a tumor suppressor and therefore a cell growth inhibitor, it is understood that PTEN will inhibit the signaling pathway to prevent tumor growth. Thus, another Western blot was performed using antibodies of proteins that are key contributors to the signaling pathway, such as PAKT and AKT. It is shown that the levels of phosphorylation were reduced by more than 50% when PTEN and PTEN isoform were used compared to their controls. This indicates that alpha PTEN negatively regulates the PI3K-AKT pathway the way that canonical PTEN does. So why use a Western blot? Overall, their goal was to figure out whether PTEN isoform was a post-translational modification of PTEN. So they used a Western blot to check if alpha PTEN is found extracellularly in PTEN protein epitope. And if it was found extracellularly, it would be detected on the Western blot to show that PTEN isoform is probably a PTM or post-translational modification. They also wanted to see if alpha PTEN decreases phosphorylation levels in the PI3K-AKT signaling transduction pathway, the way that PTEN does, to get a better idea of what its function is in tumor suppression.
We also looked at the relationship to trypsin, although the protein PTEN wouldn't cause cells to look like those treated with trypsin, cells lacking PTEN and those trypsinized would both cause changes in cell adhesion and morphology, but through different mechanisms. So visually, they would differ since trypsinized cells are rounded due to enzymatic digestion, while PTEN causes changes due to altered cell signaling pathways. The mutation or loss of PTEN is often associated with tumor formation. In relation to angiogenesis, PTEN inhibiting the PI3 kinase signaling pathway, which is a key regulator of vascular endothelial growth factor production, this prevents the formation of new blood vessels. And in relation with metastasis, the PI3 kinase pathway is also crucial for cell proliferation, survival, and invasion, and with PTEN inhibiting this pathway, it is effectively preventing cancer cells from migrating and establishing secondary tumors at distant sites, therefore inhibiting metastasis as well. In Figure 4, we have already mentioned the relationship between kinase pathway and PTEN. As PTEN acts as a negative regulator being an inhibitor or suppressor of the PI3 kinase pathway by dephosphorylating PIP3, a key signal molecular produced by PI3K, effectively blocking the activation of downstream signaling components like AKT, thereby suppressing cell growth, proliferation, and survival pathways, acting as a brake on the PI3K pathway. In Figure 5, we can see a papillary thyroid TMA spot shown in Part A is showing the deletion of PTEN. So this is lacking PTEN, which would be the opposite of suppressing. So it would be allowing for more likely for cancer cells to develop, while Figure B shows normal thyroid tissues spot showing no deletion of PTEN. which would be healthier looking cells. Here is our references. Thank you for your time.